Scientists accidentally made a mouse grow legs in the place of genitals. Turning off a gene early in mouse development led researchers to end up with an accidental six-legged embryonic mammal. This strange result took the spinal cord research of developmental biologist Anastasia Lozovska and Moises Malo to their colleagues at Portugal's Gulbenkian Science Institute in a new direction. "I didn't choose the project chose me," Malo told Sarah Reardon at Nature News. The team compared 10 to 17-day-old mouse embryos with and without functioning versions of a gene in question, TGF-BR1, which codes for the TGF-BR1 receptor protein. TGF-BR1 contributes to a signaling pathway that gives the forming body its trunk-to-tail directions. This pathway provides a creature to create a hind limb here or external genital destruction to the development embryo cells. As a mammalian embryo grows, it builds structures sequentially from head to tail. Early in development, genetic mechanisms shift from focusing on the head and extending the body and laying the foundations for the major organ systems. Later, the second transition occurs where the activation of genes in multiple layers of tissue extends the trunk to create a tail. And it's during this process that the interactions between newly merging tissues generate structures necessary for the body's exit channels and genitalia. While legs and arms share many of the same genes, this early in the process, the hind limbs and genitals have more in common. There has been some suggestion that they arise from the same initial primordial structure in ancestral species. It will be therefore interesting to determine whether a mechanism related to the developmental plasticity uncovered by our work could help explain the absence of hind limbs in snakes, but their presence in most lizards, Lozovska and colleagues wonder. The scientists found that despite the rather dramatically different placement of the extra legs in the embryos without a functioning version of TGF-BR1, the other genes expressed in these legs are similar to those found in normal mouse limbs. Both appendages arise from the middle of the tissue layers that compose an early embryo, the mesoderm. As the cells turning into the limbs push out into the surrounding outer layer of tissue, endoderm, it's possible they then receive more to become legs messages to further the development into malformed but mature limb structures, the researchers suspect. Lozovska and team took a closer look at the DNA in the mutant leg tissue compared to the control mice and identified chromatin remodeling. The proteins that control access to the cell's DNA had been switched to legs configuration instead of the genital configuration. The researchers still don't know the exact mechanism between TGF-BR1 gene suppression leading to an extra pair of legs. Understanding more about these fundamental processes will equip researchers with additional tools to tackle development changes and diseases. And the research was published in Nature Communications. This is by Tessa Koumouduras on Science Alert. Please leave your comments and thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.